My name is Claudia Vasquez, and I'll be starting my lab at the University of Washington next year. So I'm a cell and tissue biologist, and I'm really interested in understanding how our cells, what are the architectural rules that our cells use to build our organs. And this watercolor drawing that my dear friend kind of adapted from a bad doodle kind of exemplifies what I'm really interested in. Is like, how do our cells really make these structures? We're really just a series of tubes that make all these folds. And so what does it mean to be a cell that's kind of at this crux? Like, is it really squished, or are they adjusting? And how are they kind of building these phenomenal structures? And so I'm a microscopist by nature, and so what I really want to do is just watch this happen. And so what I need is a model system that allows me to watch this the whole time and at different length scales, at the organ, the tissue, and the cell, and ideally subcellular, and be able to manipulate it. You know, biologists want to break things, see if it's broken, and then finally see if we can test hypotheses of different structures. Ideally, what I want to be able to do is, if you wanted to give me any different tissue structure, I should give you 5 different ways that these cells could build this structure. And so this drove me to use the developing Drosophila renal system, the Malpighium, to use to study how they build this particular organ. And so these cells, kind of highlighted right here, start to build this organ. They're these tubes that extend 4 times in length over the course of 6 hours. And these cells are changing shape, and what you can see is that in this anterior side, in green, they form this kind of like a candy cane boot shape. And on the orange side, they kind of have this S turn plus like a hairpin turn. And so there's 2 different types of tubes that fold. And so the 2 questions that I want to really start addressing in my own lab is, first, how do these cells manage changing shape? They're changing shape throughout this whole process, and what's happening kind of in these cells that are kind of at the top versus the ones that are at this kind of like elbow turn, and how are they changing kind of the tops, kind of, you know, like the top parts of their cells versus the bottom parts of the cells. And finally, how do these cells build tubes that fold and still work? If you think of your garden hose, if you fold it in half too much, it's not going to work, and that's something that would be catastrophic for any of our organ systems. And so how are they doing this? And they do this phenomenally well throughout development and throughout our lives. And I want to leverage using live imaging, image analysis, and the Drosophila genetic toolbox. So come chat with me if you're interested in watching how things work, or if you want to talk about how cells build organs. And that's it.